Yeah, I wasn't gonna get too carried away, man.
I didn't expect otherwise. Okay, so I guess I'm going to share my screen and then try to start the slideshow. You would think that, so can everybody see it okay?
We can see you, Matt. Yep, there you go. Perfect.
Okay. I'm going to try to move a couple windows out of the way. Okay. All right. It's an honor to speak to the Nevada Native Plant Society. I can hardly believe it. I have to pinch myself to say it's true. Anyway, I'd like to thank my co-authors, Lisa Rue, Ryan Quire, Tim Seipel, who are plant ecologists, and also Dan Coleman and Eric Boyd, who are microbial ecologists. I'm not going to present on the microbial ecological data, but I'll allude to it and to the opinions of those microbial ecologists. Okay, so this photo captures the mountain big sagebrush steppe and associated vegetation at about 8,000 feet with 16 inches of annual precipitation. I'm just throwing out some numbers so you can compare and contrast the ecological variation in the sagebrush steppe. Mountain big sagebrush is distinguished by an inflorescent spikes that generally arise from a flat-topped canopy, and hopefully you can kind of see that in this picture. I'll be talking about this kind of stuff later. Okay, this area lies between the Jarbidge Mountains and the Elk Mountain area in northeastern Nevada. It harbors patches of sagebrush steppe with 100% native plant cover and high plant species diversity. Sherry Howell of the U.S. Forest Service first alerted me to this sagebrush steppe and its high quality in terms of plant diversity. What we like to think about regarding sagebrush steppe is not just plant species diversity at such sites, but also the plant functional group diversity. Different categories of plants that each include species. My phone was talking to me, I had to get rid of it. So plant functional groups, they are different categories of plants that each include species with presumed similar ecological functions. Examples of plant functional groups include species of shrubs, subshrubs, bunch grasses, parasites and hemiparasites, succulents, cushion plants, and so on. Such functional groups might have been originally well represented throughout the sagebrush steppe, but today they are perhaps not so well represented in many areas of the sagebrush steppe, particularly where the steppe has been ecologically degraded. Information bearing on plant functional groups and species diversity could allow for ground truthing the remotely sensed poor sagebrush areas, which is a relatively recent effort to conserve sagebrush steppe and the thousands of species that depend on it. And this study might also bear on the work of the late Jay Anderson, formerly at Idaho State University in Pocatello, who underscored the ecological resilience and resistance of the sagebrush steppe. For example, Anderson analyzed a long-term data set and thereby revealed how the originally degraded sagebrush steppe of the Idaho National Lab in southeast Idaho, after it was set off limits to the general public in about 1940, rebounded in native plant species and functional diversity over the following 50 years at the time he published his paper on it. The coordinates shown in this photograph are where that photograph in the previous slide was taken. So this Google Earth image shows the location of the sagebrush steppe patch captured in the opening slide. One aspect about these patches of sagebrush steppe with high functional group and species diversity is that they are tiny ecological islands surrounded by a supercontinent of human-modified landscapes. Much of the area shown in this map and on its periphery is important for livestock and cropping, in addition to having experienced increased human population growth rates. According to island biogeographic theory, these tiny sagebrush steppe patches should not exist, yet they do. Perhaps the ability of the sagebrush steppe plant functional group and species diversity, along with the associated soil and rhizosphere microbiota, is collectively is a repellent to the large population sizes of introduced plant species, which characterize well the surrounding modified human landscape. Pictured here is another small patch of sagebrush steppe at the northeast corner of the sagebrush biome in northeast Montana. This site was brought to my attention by a Montana botanist and bird hunter, Jeff Copeland. The site is similar to the Jarbidge site and plant functional group and species diversity. For example, there's about a little over 50 species per hectare at this site, as there is in the Jarbidge site. But it lies at 2,300 feet elevation with an annual precip averaging around 12 inches. This is Wyoming big sagebrush steppe, where Wyoming big sagebrush is distinguished by inflorescent spike arising from all around a rounded canopy. And hopefully you can kind of see that in this picture. So different kinds of sagebrush steppe, differing in annual precipitation and elevation, but all very similar with respect to plant diversity and function, species and functional group diversity. Okay, the scope of our study is on sagebrush steppe high and native plant diversity. And what appears to be a universal about sagebrush steppe with high plant diversity is that the winter snowpack is continuous, say from November to early April on average. And that drought is limited generally to 2 to 3 months a year. And maybe folks in Nevada might find that surprising, but that's my findings, is that's most of sagebrush steppe. A continuous snowpack and narrow windows of summer drought result in the inability of introduced annuals, including cheatgrass, to transform sagebrush steppe into vegetation with abundant introduced plants. This photo, again taken in the Jarbidge region, was taken 9 years after a fire just east of the Slide Creek campground in the Jarbidge area. Agropyrum spicatum, Castanea flava, Eriogonum heracleoides, and Lupinus arbustus represent the many forb and graminoid species common at this site. Again, about 50 species per hectare here. Notably, introduced plants are uncommon and cheatgrass is absent throughout this area, in spite of the fire. This sort of observation, where pre-fire native plant diversity is more or less equal to post-fire native plant diversity, can be made commonly throughout much of the sagebrush steppe. What is not included in our study is sagebrush steppe at the lowest elevations of the major Western North American basins, especially the Lahontan Basin, where Winnemucca resides, the lower Snake River Plains in and around Boise, Idaho, the Columbia Basin in and around the Tri-Cities area of Washington, and the Bonneville Basin, that area east of Salt Lake City. These basins have Chinook-prone winters, in other words, mild winters. Snowpack isn't persistent, in addition to about, say, 5 months of summer drought. This is where the sagebrush steppe has been transformed by humans into vegetation with abundant introduced plant species. Most importantly, it is a mistake to develop a conceptual model of the sagebrush steppe biodiversity from such sites as those that come from these low basins. And the reason for this is it can lead to overmanagement and potential harm to the rest of the sagebrush steppe. And this was a point underscored by Jay Anderson in the 1990s, early 2000s with his ecological studies. The vast majority of the sagebrush steppe resides outside the low basins, where sagebrush steppe has been and continues to be highly transformed primarily by cropping and range improvement efforts. The latter of which includes the introduction of introduced perennial grass species such as crested wheatgrass and intermediate wheatgrass. This kind of vegetation was not included in the present study because it is not plant diverse. And this is most of the sagebrush steppe. Such transformed sagebrush steppe has been highly modified for very long periods of time. Rachel Carson reported in her book Silent Spring in 1961 that the rate of overall loss of sagebrush steppe during the 1950s was about 4 million acres per year. This compares to a contemporary estimate of about 1 million acres lost per year. Regarding the history of sagebrush steppe transformation, Young and Clements underscored the significant impact that humans have had on sagebrush steppe since the middle 1800s when the Texas cattlemen brought cattle northward. So core sagebrush areas today represent about 33 million acres. That's the estimate, down from an original estimate of 250 million acres of sagebrush steppe, say prior to the middle 1800s. These are identified, these core sagebrush areas are identified using remotely sensed data, which reflect in part high sagebrush cover, high native perennial grass cover, low introduced annual grass cover, and low conifer cover. Ground truthing these core sagebrush areas and characterizing plant diversity they harbor, such as we're doing in our study, provides as a potential check on whether, for example, native perennial grasses, that second item on the list, represent a good proxy for native plant functional group and species diversity. And possibly an intact soil microbial system, which ultimately leads to the resilience and resistance of sagebrush steppe. As it turns out, and from what I know, plant diversity studies in the sagebrush steppe are essentially non-existent, and only Taylor's Sagebrush Country explicitly details in a popular plant identification guide and picture book both plant functional and species diversity found in the sagebrush steppe. He talks about the parasitic plants and the cushion plants and the shrubs and the subshrubs and all those different kinds of plant groups that are ecologically important to the sagebrush or define the sagebrush steppe today. Okay, the methods of our study. So because of the paucity of botanical research or plant diversity research in the sagebrush steppe, that in part motivated our study. Okay, the methods of our study. These include locate a patch of sagebrush steppe with high plant functional group and species diversity, and this adjacent to a road. And this often relies on the friends and colleagues such as Sherry Howell and Jeff Copeland, who I mentioned earlier. And then what we do is we run a transect 1 kilometer long and 10 meters wide in the vegetation right along the roadside, and pair that with a 1 kilometer, 10 millimeter wide transect run parallel and close to the road transect. Within each transect, we make 20 random stops and record the presence of every species within a 10 by 10 meter plot. A species found in a transect can have a maximum score of 20 if it's sampled at all 20 random stops, and a minimum score of 1 if sampled at only 1 random stop. And the point of this frequency measure is to give small annuals and plants of small sizes and statures is the equivalency of the larger shrubs and bunch grasses. Also, the idea behind the roadside adjacent to high quality sagebrush steppe is that the original footprint of the road harbors high levels of vegetation cover. And here, this road, this is Elk Mountain region in the background, Jarbidge Mountains behind us. This road that runs between the two, you can see the original footprint. The soil's more compacted, the plant composition is different than what's found out here in the sagebrush steppe. So the question is what are the plants that occupy or dominate this roadside vegetation? If roads serve as corridors for the introduction of non-native plants, then we should detect a lot of introduced plants along the road and in adjacent sagebrush steppe. If, on the other hand, sagebrush steppe plants are colonizing the original road footprint, in other words, coming from here and filling in this original road footprint, then this could indicate both the resilience of the sagebrush steppe in recolonizing the original roadside footprint and the resistance to colonization of sagebrush steppe by introduced plants that are being introduced along the road. Because of the potential high diversity of of plants along both the road and out in the adjacent sagebrush steppe. This design is biased in favor of not finding a difference between sagebrush steppe and adjacent roadside plant diversity. So that's a purposeful design. So here are the locations of 142 transects that will be reported on in this presentation. They go from up here in Glasgow, out of Glasgow, Montana, around the Lake Fort Peck area, down into the Yellowstone Plateau, down into the upper Snake River Plains of Idaho, Grand Teton National Park, the Pryor Mountain, which is the north end of the Bighorn Mountains in Wyoming and Montana, and then these down here in Elko County are the Jarbidge collections. We have many more collection sites. We have approximately 100 total sagebrush transects. So we subsampled that to get down to 68 road and 74 sagebrush transects. For total, what is that? 142 total transects. We feel that our results that I am presenting here to you guys are robust to our subsampling from a larger pool of transects. And we made this larger pool to address other questions, such as the effects of fire on sagebrush steppe plant diversity. We think that these study sites shown here on this map are representative of high-quality sagebrush steppe found throughout the sagebrush steppe biome, including in southeastern Washington, such as the Beasley Hills, which have a great collection of, or a great area of intact sagebrush steppe. Along the east slope of the Sierra Nevada, like particular sites around the Sweetwater Mountains and Walker River drainage, and possibly sagebrush steppe around Gunnison, Colorado, where the Gunnison sage grouse, large populations of Gunnison sage grouse are centered. Okay, so for results, our results include that most plant functional groups, which we were interested in a priori, each showed little species diversity differences between sagebrush steppe transects and those along adjacent roadsides. Although likely an artifact of our study design, the high plant functional group and species diversity along roadsides adjacent to high quality sagebrush steppe might be taken as an indication of ecological resilience. High quality sagebrush steppe has the ability to colonize the original footprint of the road. That is, if the sagebrush steppe adjacent harbors high levels of native plant diversity. For example, nitrogen fixers, including introduced and native legumes, were equally diverse along the roadsides and in adjacent high quality sagebrush steppe. This photograph of the Astragalus whitneyi variety confusus comes from the Elk Mountain area in Elko County, Nevada, and this species could be found along the road, but mostly in the sagebrush steppe. And this is true of, and I'll just point out, I won't point out all these, but I'll just emphasize shrubs and rhizomatous native groundworts, just to illustrate this point. Okay, so about functional groups that don't differ between sagebrush steppe and adjacent roadsides. Okay, so I'm going to have to explain this graph here, but so notice that I'm going to talk about shrubs. So back to this previous slide, shrubs don't differ between sagebrush steppe that appears pristine and adjacent roadsides. We could take a look at, so there's 42 shrubs sampled in this study, and this y-axis represents Shannon Diversity Index. Shannon Diversity Index accounts for species richness and evenness. Okay, so the y-axis is the Shannon diversity of the shrubs for all 42 species. The x-axis represents categories of roadside. Here, 68 transects. And sagebrush steppe transects, 74 of them. The filled circles represent a transect. So roadside transects here, sagebrush steppe transects here. Circles with high values represent transects, or filled circles with high values represent transects, in this case, roadside transects that have high shrub diversity. Whereas transects with down here at the bottom, 0, represent transects in the sagebrush steppe or in the road where there was maybe one shrub species, so no diversity. Okay. So that's just to give you a feel for this general category of no difference among many plant functional groups. One group of that we were that surprised us were we sampled a total of 17 native rhizomatous graminoid species. Not only did they not differ between the sagebrush steppe and the roadside. Oh, I forgot to point out one thing here, is these triangles are the mean values, the mean diversity values for all of the roadside, or the mean diversity values for all of the sagebrush steppe. So the mean values aren't really different. And also what I forgot to point out is that this 95% confidence interval, if there's a negative and positive value, that means the confidence interval bounds 0, which means there's no significant difference between the two. Okay, so that was the same for the shrubs. A negative and a positive means not significantly different. Sorry about that. Okay, so here we're back to rhizomatous graminoids. They don't differ in their diversity between roadside and sagebrush steppe, but overall they're hardly present in sagebrush steppe where diversity is high. And you know, the two most common native rhizomatous graminoids were thick spike wheatgrass and western wheatgrass. I call them Agropyron daisystachyium and Agropyron now I'm having let me just move this out. Agropyron smithii, daisystachyium with the hairy spikelets with hairy lemmas, and smithii with the glabrous lemmas more. Two very similar native rhizomatous grasses that are not that common. When they are common, they seem to be a roadside, but also creeping into the sagebrush steppe. And we just wonder if maybe the rhizomes, even when produced by native species, maybe they can't compete with an intact sagebrush steppe ecosystem that has an intact soil microbiota. I don't know. That's just a working hypothesis. That's why I wanted to point out native rhizomatous graminoids being really uncommon, not commonly sampled. All right. Of the functional groups showing, let's see, so plant functional groups with greater species diversity along roadside than in adjacent sagebrush steppe, it was really only basically one functional group, and that is the introduced plant species. We sampled a total of 648 species in this study in these 142 transects. Of these, 98 or 15% were introduced plant species. Introduced plant species are those that arrived in North America due directly or indirectly to European and later immigrations. They have been selected to optimally perform where disturbances are regular or impactful, so they are ecologically similar in this regard. Introduced plants, or any subset of this functional group, including individual species, I've listed some subsets here of introduced plants, are the only plant group to be more diverse or abundant along roadsides. So here is species diversity of all 98 introduced plants is significantly greater. And here indicates the confidence interval, both right here, this 99.5% confidence interval. It doesn't include 0, both numbers are positive. So these values here, this triangle over here in the roadside transect and this triangle over here in the sagebrush steppe, among the sagebrush steppe transect, that value in the roadside transect is significantly greater than the value in the sagebrush steppe. So this is somewhat surprising because these 98 species are super abundant in adjacent croplands and human transformed rangelands that surround these sagebrush steppe study sites. The same finding was made for a subset of invasive or noxious plant species. All 30 that we sampled are state and county listed introduced forms. For a group of 30 plant species, and again, you can see the value here that the two values, the confidence intervals are both positive. So this mean value here is significantly greater than this mean value over here, roadside greater than sagebrush steppe. So for a group of 30 plant species that have a reputation for being highly aggressive colonizers, they are overall not so diverse along the roadside or adjacent sagebrush steppe transects, suggesting that the entire system that we sampled is fairly resistant to colonization by so-called noxious weeds. Or it could mean that noxious weeds have an undue reputation for being aggressive colonizers. The same finding was made for individual introduced species, such as crested wheatgrass. Here, the y-axis, now I'm dealing with a single species, not a functional group of species, but a single species, in this case, one that comes from the functional group introduced plants, Agropyron cristatum, crested wheatgrass. Its frequency. So here the y-axis is not Shannon diversity index, it's the number of times sampled in a transect. So these transects up here, crested wheatgrass was sampled at all 20 random stops. These down here, crested wheatgrass was not sampled at all in these transects. And you can see that on average, the roadside has a lot more crested wheatgrass than do the sagebrush steppe sites. And this is really only pronounced at sites that are drier. Precipitation ranges from, say, down here 200 to over 600. So down at the most red is about less than 10 inches of precip a year, and the green is over 20 inches of precip a year, just over. Okay, so the model here is, yeah, it does matter whether you're in the roadside or the sagebrush steppe for explaining the abundance of crested wheatgrass, and it matters most if you're the dry site rather than the wet site. Okay, so that's crested wheatgrass, a subset of introduced plants. And the same with cheatgrass, and I've got to mention cheatgrass because it has such a bad reputation, maybe deservedly, even though humans purposely introduced it. Still. So the same findings as I explained to you before with other introduced plants and functional plant groups of introduced. Same finding was made for cheatgrass. Sites with higher precipitation tended to have little or no cheatgrass. So there is a significant difference between roadside cheatgrass frequency and sagebrush steppe cheatgrass frequency, even though the numbers appear all over the map. There's a lot of sites in the sagebrush steppe down here that weren't sampled for cheatgrass, and these particularly come from the wetter sites. And the same is kind of true for the roadside sites, although there's greater frequency and more roadside transects. Regardless of these findings, the sagebrush steppe sites with a high frequency of cheatgrass, so that would be these over here, were found in the upper Snake River Plains, say the Idaho National Laboratory, between Arco and Idaho Falls, where summer drought averages more than 2 months. So here's a picture from the Idaho National Lab of the sagebrush steppe, and you can kind of see a faint hazy purple. A lot of that is cheatgrass. It was sampled in all 20 random stops in this particular transect here. However, at these sites, plant functional group and species diversity was high as anywhere in the sagebrush steppe. You can see paintbrush here, a hemiparasite. You can see bunch grasses. There's a squirrel tail and different species of stipe, and there's different forbs. There was again on average there was about 50 species per hectare or per transect in this area too. So this would indicate that in high quality sagebrush steppe, even if cheatgrass gets in, because say the summer months are droughty, more than 3 months or 3 or more months, that it still doesn't have an effect on the native plant diversity. And incidentally, when fire comes into the Idaho National Labs, which it has done, unfortunately, very frequently, it removes the cheatgrass for the 15, 20 years I've been observing this area. Okay, so let's see. So now we're in the final part of this talk, and that is what were the plant functional groups that had greater diversity in the sagebrush steppe compared to the roadside? And I'll point out some of these: native perennial grasses, subshrubs, cespitose perennial forbs. These are the cushion plants, the cushion eriogonums, the cushion sandworts and such, the tuber-producing plants such as calochordus and allium species, the ancient North American legumes. These are the legumes that, like the New World astragalus, Dahlia, Ceralia, where genetic evidence suggests they are paleo-endemics. They've been evolving in North America for at least the last 4 million years, if not longer, and they have lots of endemic species. Perhaps they've had time to associate with the soil microbial system compared to many other legumes that are absent from the sagebrush steppe. And then the parasites and the hemiparasites, succulent, and then ferns. I would say dry adapted ferns and spike mosses. Those are also in the sagebrush steppe and rare along the roadside, according to our study. So here's some results. I'll just illustrate a couple of these. Here is on the y-axis here is Shannon diversity of the 58 native perennial bunch grasses. So this would be considered a functional group. In this case, the 95% confidence interval, both numbers are negative. So again, it doesn't encompass 0. So that means this mean value in the roadside is significantly less than this mean value in the sagebrush steppe. So bunch grass diversity is higher in the sagebrush steppe. Then precipitation provides a lot of this R squared higher, relatively higher squared value, in that most bunch grass diversity is occurring at the wetter sites. You can see all the green transects are occurring higher up in each column. Okay, but there is a difference between roadside and adjacent sagebrush steppe with respect to perennial bunch grass diversity. And I'm going to emphasize bunch grass diversity because this project involving the identification of core sagebrush areas relies on perennial grass diversity, not perennial bunch grass diversity. And then the perennial cespitose forbs, the cushion plants, like this sandwort, Aramagonia, I still call Arenaria franklinii, Eriogonum ovalifolium, Eriogonum cespitosum, and so on. There was a total of 62 perennial cespitose forb species sampled in this study. You can see that many roadside transects did not have any, and the 99.5% confidence interval does not encompass 0. They're both negative, so that means this mean value along the roadside is significantly less than that mean value in the sagebrush steppe. So there tends to be a big difference between perennial cespitose forb diversity in the sagebrush steppe compared to even in the adjacent sagebrush or adjacent roadsides, which are pretty rich in native plants. And there tends to be only a modest contribution of precipitation. For example, it appears that in the roadside transects, the perennial cespitose forbs tend to be more diverse and drier, generally lower elevation sites. And so I could go on and on with those graphs, but I'll spare you the details. I'll just point out these other that I've already alluded to, these other plant functional groups. So you say the nitrogen fixtures, the nitrogen fixtures that belong to the legume family that show genetic evidence of being Paleo North American endemic. So lots of endemic species in North America for these kind of astragalus. The parasites and hemiparasites such as Castanea flava, which was one of the species of Castanea common in the sagebrush steppe of the Jarbidge area. Subshrubs like Eriogonum microthecum. Succulents such as Coryphanthus species, the vipera shown here. Missouriensis was also sampled, as was Pediacactus and Polycanthus. Then Woodsia oregana. It wasn't sampled that much, but when it was sampled, it was in sagebrush steppe, never roadside. The same with spike mosses, Selagina oleodensa. And so, if we combine, in this slide here, the y-axis again is Shannon diversity. And what I've done is combine all these groups. All these plant functional groups pictured here, plus bunch grasses, plus cespitose forbs, into one large group that range ecologists might be referred to as the functional group decreasers. Decreasers are plant species that become less abundant with greater rates of grazing and trampling. So if we do that, there's 187 total. And you can see that it's a 99.5% confidence interval. Both numbers are negative, which means this mean value in the roadside is significantly less than this mean value in the sagebrush steppe. So the decreasers are definitely more common in the sagebrush steppe than the adjacent roadside. But there's still a relatively high diversity in many roadside transects, which would suggest to me that this decreaser group that I've combined over here has a pretty good ability to recolonize the original footprint of the road. In other words, sagebrush steppe is resilient with respect to these decreasers. Okay, one last data slide before the second. Because the identification of core sagebrush steppe areas is focused in part on native perennial grasses, we have presented evidence that perhaps should be modified to focus on native perennial bunch grasses. Presumably, because the core sagebrush areas involve remotely sensed data, presumably a bunch grass growth habit is amenable to remote detection. For example, I can personally detect bunched graminoids at 80 miles an hour. That means they should perhaps be detected somehow remotely. Okay, these two scatter plots both have the same y-axis. That y-axis is the Shannon diversity of 58 native perennial bunched graminoids. So I'm taking all graminoids, bunched grasses and bunched sedges and whatever, all the graminoids. There's 58 of them. So you can see that's on the y-axis here, that's on the y-axis over here. The x-axis on the scatter plot to the left represents the diversity of all 550 native plants that were sampled in these data I'm presenting to you. The x-axis in the right scatter plot represents the diversity of all 98 introduced plant species made in the 142 transects. To the left scatter plot, native perennial bunch grass diversity is a function of total native plant diversity. So this is a function of this. With higher levels of bunch grass diversity in wetter sites, the larger open circles. The smaller open circles are drier sites, the larger are wetter sites. There's the key over here, 200 milli. So going from 10 inches to 20 inches a year, more or less. To the right-hand scatter plot, native bunch grass diversity is not, so native bunch grass diversity here is not explained by introduced plant diversity. In other words, although native bunch grasses are still most diverse in wetter sites, they're not, that precipitation is explaining some pattern over here, not introduced plant species. This suggests that native bunch grasses can coexist with a diversity of introduced plants. This left-hand graph suggests that native bunch grass diversity can serve as a proxy for all native plant diversity in sagebrush steppe, and perhaps native perennial bunch grasses can be used as in remotely sensed data to identify poor sagebrush areas. The right-hand graph suggests that native bunch grass diversity is ecologically resistant to introduced plant diversity. The findings I present here can be repeated for all those plant functional groups that I put into the decreaser assemblage. But the important point here is that overall results are not as strong when native perennial grasses are on the y-axis instead of native perennial bunch grasses. Okay, to summarize, high-quality sagebrush steppe might be detected, say, in ground truth in core sagebrush areas by simply knowing general and potentially coexisting plant groups, like, are the plants I'm looking at, are some of them native perennial bunch grasses? Although for native perennial bunch grasses, one would have to know what Agropyrum cristatum is and exclude it because it's an introduced bunch grass, perennial bunch grass. Simply being able to identify a subshrub like those in the genus Eriogonum, or cushion plants like those in phlox, or tuber-producing plants like those in Allium or Californis, legumes in general like astragalus or dahlia, or hemiparasites like bastard toadflax or paintbrush species, cactus species, certain ferns and spike mosses. So identifying high quality sagebrush steppe could be easy from identifying the plants to general categories rather than species. Finding just one common species from each of these plant functional groups would indicate high quality sagebrush steppe with perhaps an intact soil microbial community that could resist introduced plant colonization and potentially be resilient and thus expand into degraded adjacent rangeland. And as one final note, I would like to say that all these findings that I report are simply restatements of all those made by J. Anderson and colleagues 25 to 30 years ago. Thank you for your time.
Thank you. Yeah, another round I can hear this in the music. Thank you very much. Has anyone got any questions? Are there any questions from the room?
Thank you, Matt.
You bet.
Are there any questions online in the chat?
We don't have anything in the chat yet.
No.
Matt, this is Marcus. I had a question for you.
Hi, Marcus.
So I was looking at the Shannon diversity graphs you had, and I was focusing specifically on precipitation. Now, in some of them, you pointed out that there was a correlation between the precipitation and diversity values. But in a lot of them, like I was looking at the shrubs and the rhizomatous graminoids, there really wasn't. So I was wondering if there were, I don't know, across the board, if you're looking at all the functional groups, which ones, I guess, of the natives did you find a correlation with precipitation?
So as I tried to point out the couple instances where precipitation was most profound, like in bunch grasses where bunch grass diversity is higher with higher precipitation. Cespitose forbs perhaps being more diverse where precipitation is low. But in a lot of them, the precipitation was contributed marginally to the model. And it was kind of a mix about whether I should present it, but there was the few instances where I thought it was worth pointing out, like crested wheatgrass and the cheatgrass are definitely abundant where it's drier. And when precipitation is above, say, I don't know what a threshold might be, but let's say 15 inches a year, there should not be crested wheatgrass anywhere. And if there is, it's because somebody's been rampantly spraying herbicide or doing something to upset the natural ecology of that species and make it abundant. And that happens in Montana all the time. There's lots of cheatgrass, but it'll go away if people just leave it alone and quit spraying it. So as a consequence, it's found commonly around crop fields. I guess I'm hopefully I'm answering your question. I guess I'm trying to say is that precipitation, it mattered here and there, but I tried to emphasize where it was most important.
Sure. I guess I was just thinking, I guess I was thinking across the whole board, but then also specifically, your Woodsia and your Selaginella. Was there any sort of pattern there?
Yeah, and the Woodsia ranges from sites in Montana to down in the INL, which are the summer droughty, like in the Craters of the Moon National Monument. I had study sites there with Woodsia, and there were sites in this Yellowstone Plateau with Woodsia. Those are two very different. One's dry, droughty, and another is wet within the realm of Maya, within the climatic envelope I'm dealing with.
Interesting.
Yeah, and spike moss, I'd have to say spike moss is the same. It's in eastern Montana where it's low and 12 inches a year, but it can be in mountain big sagebrush with over 16 inches of precip a year.
Sure. Did you ever look at, comparison instead of precipitation, maybe like elevation or like other climatic variables?
Yeah. I was just cutting to the quick. I did a lot of variable selection over the years. Yeah, and it seems like annual precipitation always comes out is the most important.
Okay. Gotcha.
Yeah.
Thank you.
Yeah, I glossed over that. Sorry.
That's all right.
Well, thank you again. I don't think we have any questions in the room here. Sarah, have anyone online got questions?
Nope, nothing in the chat.
Well,
Okay.
I'd like to thank you again for a wonderful presentation.
Yeah,
I'd
thank
also
you.
like to remind all members to do check your email. I'm not quite sure when it will come out, but for the ballot, because the voting will take place online, and we will announce the results at the November meeting, which is on November 6th. And the presenter for that will be Tiffany Piera talking about the art of science. So another round of applause and thank you. And thank you very much for participating.
Thanks, Emma. Thanks, everybody.
Thank you, Matt.
All right. Take care.